Hi students, I am Sagar Kumar, MSc PhD. Now in this video, most important video it is because many questions are arising are possible to arise from this video in the NEET. How? Now you see in this video I am going to discuss mainly three subheading, that is the respiratory balance sheet, and second one is the amphibolic pathway and respiratory coefficient. All these three are present in the NCERT book, and some mathematical means calculated mathematical also, some problems also will be created from this one. If you know the concept of this one, you can solve any type of the question. Complicated questions can be arise from these three subheadings. So please kindly properly listen and see this video and try to get more maximum marks in the NEET. Now first one we will see respiratory balance, the respiratory balance sheet. The respiratory balance sheet shows how many ATP molecules are produced from one sugar molecule, because the sugar molecule used to follow in different steps, that is the glycolysis, next followed by the next reactions will be in the mitochondria. So different steps, different number of the ATPs are produced, that all of you know, that we are going to see in this one. Now why, how it is it will be done? Actually it is a theoretical and assumptional. Why? Because theoretical means here, whenever the glucose molecules oxidizing in the different steps, glycolysis, and next in the mitochondrial matrix, they can be withdrawn. Whatever the intermediate products are there, they can be withdrawn to produce proteins or the fats. Or proteins or fats are may join by the breakdown into this. This is natural, it is just to happen generally in the living cell. But it is, even though it is in the stepwise and organized, and each is having its own number of the ATP molecules production, but it will be done. It is cannot be done like that. So we are calling it as theoretical. Theoretically, it is possible, as stepwise it is there, and assumptional, we are assuming how it is. Now, I discussed that the sequential means, because first step will be the glycolysis, and then in the final in the glycolysis end product is the pyruvic acid. It will go to the mitochondria. Again, it will take the link reaction, Krebs cycle. In all these steps, it is to produce direct some ATPs and energy stored molecules also it will produce. They are the NADH plus and FADH2. And these are the energy stored molecules. This will be converted into the ATP in the electron transport system. Now, the thing is, how many they are producing in each, and if the NADH is there, how many it will be converted, it will give rise the ATPs. All the things we are going to calculate and show in a sheet, that is the balance sheet. Now here he has given some 4 points. Those 4 points also very important for the NEET exam. First point, this is a NADH synthesizing places where it will be produced, it will be produced in the glycolysis and a link reaction and a Krebs cycle. And next, intermediates, whatever the intermediate products means, whatever the so many, nearly 20, 25, 20 intermediate products are coming, they may convert into some proteins like this, in this whatever amphibolic pathway they may go, or the fats and proteins, they may be break down and they may enter into the in this cycle. So balance sheet is not possible because of that one. But theoretically, assumptionally, we can calculate, but it is not practically it is not done in the living or any living cell. That only he has given. This is the most important concept. For the why it is theoretical, or assumptional. Means the intermediate compounds whatever are produced during the glycolysis and mitochondrial matrix link reaction Krebs cycle may be withdrawn to form the proteins or the fats, or the fats and proteins are oxidized, they break down and they may enter into the this whatever the respiration. So it is practically it is not correct, but assumptionally theoretically we can calculate how that we are going to see. And now you see here one more thing he has given, the differences between the fermentation and aerobic respiration. This is most important, but many people are neglecting. But he will give in the statements, remember it. What are they? Fermentation and aerobic respiration. Fermentation and aerobic and fermentation means anaerobic respiration. Anaerobic respiration without oxygen. So in this one, fermentation, partial oxidation is done in the fermentation. Means glycolysis, whatever the partial oxidation has done, that is only the thing, and 2 ATP molecules only will be produced from the in the fermentation. And next, complete oxidation will be taken place in the aerobic respiration by involvement of the glycolysis, link reaction, and a Krebs cycle. So it will produce 38 ATP from one glucose molecule. Only 2 ATP molecules will be produced in the fermentation as it is partial oxidized. And next, if you see in the aerobic respiration, whatever the energy rich products will be produced in the link reaction and Krebs cycle and glycolysis, they will be converted into the ATP in the electron transport system. And 38 ATP will be produced in this one, first difference. And next will be the NAD fermentation to ATP, and 38 ATP will be produced in the aerobic respiration. And next will be the complete oxidation, partial oxidation in the fermentation, complete oxidation in the aerobic respiration. Third point he has given, this is the most important point, but so many people are neglecting, remember it. That is, NADHH plus is produced in the glycolysis when glyceraldehyde 3-phosphate is converting into the biphosphoglyceric acid. At that time, that glyceraldehyde 3-phosphate will be oxidized, NAD plus will be reduced into the NADHH plus. These are the neat questions. So that NADHH plus which is reduced in the fermentation, it will be reoxidized. That you already studied. Same thing, NADHH plus will be oxidized in the electron transport system in the mainly two compartments, that is the complex number 1 and complex number 2. It will be oxidized. NADHH plus will be oxidized in the complex number 1. FADH2 will be oxidized in the complex 2. But it is difference he has given very clearly in the textbook. Please remember it. It is the most important for the NEET. That is, NADHH plus is oxidizing in the two places. How many places? If we ask, 2 places in the respiration, fermentation 1 place, and in the electron transport, that is one NEET question. And is there any difference in the oxidation speed? Yes, the difference he has given in this. That is, NADHH plus will be oxidized very slowly in the fermentation. That is the formation, and at the time of the formation of the ethanol, or acid, that is the lactic acid formation. This is the thing. He has given very clearly in the textbook. Such type of bits only will catch and then it remember it. So that, but in the oxidation, in the electron transport, this is very vigorous, very high speed it is. But fermentation, NADHH will be slowly oxidized. That is most important point, remember it please. Now we will see how many are ATP are produced or energy stored molecules are produced at different steps of the aerobic respiration, which is to be held in the stepwise. The first one, whenever we are seeing glycolysis, ATP. In the glycolysis, already studied very nice. 4 ATP are produced and 2 are consumed and 2 are the net ATP. So here, whatever where it is consumed and where it is produced, all of you know. 3 carbon compound, that is the glycerol 3 phosphate and NAD. The next is the DHF dihydroxyacetone phosphate. Both they will produce 2 each. NADHH plus will be, and this one, whatever the glycerol 3 phosphate and dihydroxyacetone phosphate will be converted into glycerol 3 phosphate. So they will produce in that way. 4 are produced but 2 are consumed. Net gain is 2 ATP molecules. And one more thing you must remember here, all the 4 ATP molecules are produced through the substrate level phosphorylation. This is most important question. But he mentioned first substrate level phosphorylation in the Krebs cycle only in the textbook, but when he is writing about the Krebs cycle, but it is applied for the glycolysis also. If we ask how many ATPs are produced in the substrate level phosphorylation in the aerobic respiration, how many are produced? Means produced are 6, 4 in the glycolysis and 2. But when you are taking it into the 38 count, at that time it will be counted 2 only, because our 2 are already 2 are consumed in the glycolysis. This concept, if you knows, he can ask the question in different ways. 6 are produced substrate level phosphorylation, but glycolysis 2 are consumed, so 2 are the net, and 1 will be produced, 1 NADHH plus will be produced. Then the glyceraldehyde 3 phosphate is converting into biphosphoglyceric acid. The glyceraldehyde 3 phosphate will oxidize, and NAD will be produced and forms 3 carbon compound 1. So glucose gives a 3 to the dihydroxyacetone phosphate and this one. So both will produce it will be the 2 NADHH plus will be produced in the glycolysis from 1 glucose molecule, and ATP net will be the 2. And now the end product of the glycolysis, which is the pyruvic acid, will be transported into the mitochondria, and it reaches into the mitochondrial matrix. In the mitochondrial matrix, before the pyruvic acid is going to enter into the Krebs cycle, it will take one more reaction that we used to call it as a link reaction. Not in the textbook, but we used to call it as a link reaction. In the link reaction, before entry of the pyruvic acid into the Krebs cycle, one reaction is there. In that reaction, that is a decarboxylase dehydrogenase reaction, we can say. And dehydrogenase means here it is to lose one hydrogen and it will be added to the hydrogens will be the NAD plus, and NADHH plus will be produced from one pyruvic acid. So 2 are there, 1 plus 1. 2 pyruvic acids will come from one glucose molecule. So 2 are produced there in the link reaction, NADHH plus. Now, and NADHH plus link reaction, there is no question of formation of the ATP or other things. Only NADHH plus will be produced, and 1 carbon dioxide will be sent out. That is also important for the need, but it is here it is not necessary. Next, Krebs cycle, the acetyl coenzyme A will enter into the Krebs cycle. The acetyl coenzyme will be condensed with the oxaloacetic acid by taking the one water molecule, and citric acid synthesis will be used, and it will be converted into the citric acid, isocitric acid. Isocitric acid will be oxalosuccinate. There one NADHH plus will be produced, and alpha ketoglutarate, when it is oxidizing the decarboxylase and dehydrogenate, there one NADH plus will be produced. And next malic acid, when it is converting into the oxaloacetic acid, one more NADH plus will be produced. So in this way, in the Krebs cycle, 3 NADH plus will be produced from one pyruvic acid. Such type of 2 pyruvic acids are there. So 3 plus 2, 6 are produced. And FADH2 will be where it is produced in the Krebs cycle only. When the succinic acid, when it is converting into the fumaric acid, then only it will produce one FADH2. So two pyruvic acid molecules. So it will be the 2 FADH2 will be produced from one sugar molecule. Now we will count the one molecule of the GTP, which will be immediately converted into the ATP, will be done in the Krebs cycle. That is when that succinyl coenzyme is converting into the succinic acid. At that time, one GTP will be formed. That is ATP equal to ETP. So pyruvic acid from one and one glucose, 2 pyruvic acid will come from glucose. So 2 ATPs are generated in the Krebs cycle. Now we will bring all these things into a balance sheet. That is, in the first we will see NADHH plus. NADHH plus are produced in the glycolysis 2 and the link reactions 2 from one glucose and Krebs cycle 6, total 10. So all of you know that is the electron transport system has given one NADHH plus will give 3 ATP. So one NADHH plus as giving it is 3 ATP. 10 NADHH plus will give how many? 30 ATP will be produced. This is the most important bit. How many NADHH will be received by the complex number 1? That also, if you see neat questions, you can ask anywhere. And next, FADH2 will be produced 2 from one glucose molecule and they will produce 4 ATP because each one will give 2 ATP, 2 into 2 it will be 4. Now you see here, this entire thing is produced in the electron transport system. This also will ask. How many ATP molecules are produced in the electron transport system? 34 out of 38. And what about the 4? The 4 will be the 2 plus 2. That is the total. That glycolysis 2 are produced. And next will be the Krebs cycle 2 are produced 4 ATP. These 4 ATP are substrate level phosphorylation. That is also important. You see, if you understand the properly, it is nothing. You can give answer for anything. So total ATP which are produced are 38 ATP from one glucose molecule in the aerobic respiration. If it is anaerobic respiration, it is only the 2 already we discussed in the differences. This is the balance sheet. And if you want to note down all the things on one place and read it properly, you won't get any doubt and any type of the calculations or any problem he gives also, you can solve it very easily by knowing this one. Okay, now we'll see amphibolic pathway. This is also very high, most important. But some majority people are leaving that. But he can give very nice, very critical questions if he can create from this one. That I am going to show you. What is amphibolic pathway? Amphibolic, amphibians means they can live in the water and on the land also. For the amphibian means if anything is acting for the two places, two things, it is amphibolic. Here, whenever we are talking about the photosynthesis, it is anabolic. And respiration, catabolic, all of you know. But whatever the respiration, it is not a catabolic, anabolic also. He want to prove that in this amphibolic pathway. How it is? Because Respiratory substrate, whatever you studied in the glycolysis with glucose or fructose, we have started and we saw. And at the same time, in the respiration, he has given that is one thing in the respirator substrates, carbohydrates, proteins, and fats. Other two also there. So they won't enter directly from the beginning. They will join in the middle way. That only we told here. That is the sequence. They will be intermediate products will join in this cycle. That is the glycolysis link reaction. Somewhere it will join. So the breakdown of the fat or the proteins will join in the whatever the respiration sheet. Somewhere it will join in the respiration. That is the glycolysis. It may join in the glycolysis. It may join in the link reaction. It may join in the Krebs cycle. How it will be that we are going to discuss in this one, most important one. Now, all of you know that carbohydrates are the products of the photosynthesis and products. So, in the carbohydrates, whenever we are seeing, carbohydrates will become simple sugars. That is the glucose is the sugar which is favored substance for the respiration. He has given what is the favored, why it is. Did you think about it? Favored means here the only the glucose molecule will completely pass through the entire respiration pathway. How? Because glycolysis 10-step chain reaction will be done by the glucose only. Then next link reaction to whatever the pyruvic acid molecules is done, Krebs cycle, and it will enter again into the electron transport system. So in that way, glucose is favored. Glucose is a stable molecule. I discussed that one in the glycolysis also. And glucose is the end product of the photosynthesis and it will be converted into the sucrose. So first product is the glucose only. So all the living organisms are adapted to take the glucose as the favored substract. That is the meaning. Now the whatever the glucose will be converted into the glucose 6 phosphate. Glucose 6 phosphate will isomerize and it will become fructose 6 phosphate. Fructose 6 phosphate will become fructose 1,6 bisphosphate by taking 1 ATP. All the things you know. And the fructose 1,6 bisphosphate will be splitted. It will be break down. It will be break down into 2-3 carbon molecules. They are the dihydroxy acetone phosphate, DHAP, and glyceraldehyde 3 phosphate. Two are produced. And from here, this finally glyceraldehyde 3 phosphate will run further 5 more reactions and forms the 2 pyruvic acids. And the pyruvic acid will enter into the mitochondria, and in the mitochondria the link reaction whatever we told here, Krebs cycle will be taken, and here it will be oxidized fully on the entire energy which is present in the glucose will be released. This is the way. Now we are going to see when how the fats will be involved in the respiration, how the proteins will involve in the respiration, not only amphibolic pathway. Now you see here the fats. Whenever you are seeing the biomolecules, fat, that whatever the lipids are made up of by the fatty acids and glycerol. Glycerol and fatty acids both are present in a lipid. We are calling generally fats only. That all of you know. That only has given. The fats contains of fatty acids and glycerol. Glycerol is 3 carbon compound. Remember it. Fatty acid simple fatty acids up to the 19 can be present in the R group he has given. So 20 with carboxyl in the simple, but complex compound between many will be maybe there. But fatty acid, one fatty acid will be attached to the one hydroxyl group of the glycerol, which is called the 3-hydroxypropane. Trihydroxypropane it is. Now, first, the fats will be break down into the fatty acids and glycerol. The glycerol will convert into the dihydroxyestone phosphate. That is also 3 carbon, this is also 3 carbon compound. But the thing is, glycerol C3H8O3. But that 2 hydrogens it will do something, it is not necessary, no. So dihydroxyestone phosphate, glycerol will be converted into, to enter into the respiration, glycerol will be converted into which one? Like that also you can ask in the NEET. It will be converted into the dihydroxyestone phosphate. The dihydroxyestone phosphate can be the glyceraldehyde 3 phosphate, both are the isomers. So the dihydroxyestone phosphate will be converted into the glyceraldehyde 3 phosphate. So glycerol will be converted finally glyceraldehyde 3 phosphate via dihydroxyestone phosphate. Now we calculate how many ATP will produce. Yeah? Huh? No. Just to calculate, 20 ATP it will produce. One glycerol will produce 20 ATP. How? From here, come down here, glyceraldehyde 3 phosphate will be it will become pyruvic acid. Whenever it is converting to the pyruvic acid, it will produce 2 ATPs. Glyceraldehyde 3 phosphate, when it is converting the biphosphoglyceric acid, phosphoglyceric acid, at that time 1 ATP, and 5 PEP, phenol pyruvate, 2 pyruvic acid, 1 ATP. So 2 ATPs directly produced and 1 NADH is produced. Glyceraldehyde 3 phosphate is converting to the biphosphoglyceric acid, at the time DHAP will come now. That DHA, dihydroxy, that is NADH plus. NADH plus will produce. NADH plus will produce, give you how many? 3 ATP, and produced are the 2 ATP. So by glyceraldehyde 3 phosphate, when it converted into the glycerol converted into the dihydroxyestone phosphate to glyceraldehyde 3 phosphate, it will produce 5 ATP before converting into the pyruvic acid. This you will ask in the NEET. You should know how it is. In this one, when it is coming, there is no losing of the ATP, because glucose, when it is converting all the things at the time glucose, but directly it is forming the glyceraldehyde 3 phosphate. So here 5. And next in the Krebs cycle, whenever we are seeing in the Krebs cycle link reaction, pyruvic acid in the link reaction, 3 ATP will be produced as we shown. That is a link reaction. 2 will be produced, 2 pyruvic acid, 2 will be produced. So here the thing is 1 pyruvic acid will give 3 ATP by forming the NADH plus in the link reaction, and in the Krebs cycle 12 ATP. How 12 ATP? That is, here you see, for that one Krebs cycle 6 are formed, NADH plus and NADH plus, this is the 2, and the next will be the 1 plus 1. Total how many? That is, in the Krebs cycle 3 plus 6, 6 into 3, 18. FADH2 will be the 2 means 1. Now the thing is, 1 pyruvic acid will produce 12 ATP, means 24 they are producing in the Krebs cycle by 2, 12. 12 plus 3, 15. 15 plus 5, 20. That only I shown. This he will ask. Remember it. Many people, I don't know, but I saw many people, they are not showing this because he will catch such type of points. Please remember it. Now, so glycerol will produce when it becomes the dihydroxyacetone phosphate. How in the respiration, how many ATPs it is able to produce? He will ask. Now come to the fats. Fatty acids are having so many number of carbons, but the fatty acids will be beta oxidation will be taken, and it will be converted into the acetyl coenzyme A. Acetyl coenzyme A, 2 carbon. Fatty acid contains 20 carbons. How many acetyl coenzymes A it can form? He can ask, because it is present in the chemistry also in our NCERT. So, if it is fatty acid is having this many carbons and it produces this how many number of the acetyl coenzyme A, and that many number acetyl coenzyme A produces how many number of ATPs? Simple. Beta oxidation is not mentioned because beta oxidation also ATP will produce. But he is telling this many 20 are there arachidonic acid. It form the acetyl coenzyme A, and acetyl coenzyme A, after forming the acetyl coenzyme A, arachidonic acid, after forming acetyl coenzyme A, how many ATPs it produce? What? 10. That is the 20. 20 by 2. It is acetyl coenzyme, the 2 carbon. So 2 carbon, 10 will be formed. 10 into 12, 120. Over. And he can critically can ask. Whenever the fatty acid is, that I will tell you next. This is next is proteins. So if the fatty acids are coming, it after converting to the acetyl coenzyme A, it will produce 12 ATP. If the glycerol is coming, it will produce 1 DHAP will be produced 20 ATP. And proteins, whenever they are coming, proteins are made up of amino acids. So that will be break down into amino acids. Amino acids will be again break down and it will forms different carbons compounds. Out of that one, they will produce pyruvic acid, which is 3 carbon compound. Pyruvic acid will be formed. So one amino acid, whatever it may be, amino acids form the pyruvic acid. After forming the pyruvic acid, that pyruvic acid will produce how many ATPs? 15. Over it is, because link reaction 3 and krebscycle 12. It is 15. But that only I wrote here. Now, we came to know that fats and proteins will be break down and they may enter in the respiration in the different places of the pathways. So, when the way is like this, it is not one-way traffic. When it is coming like this, acetyl coenzyme A cannot convert into the fats. Yes, it can. When it is coming like this, it may form this. And glycerol, whenever it requires dihydroxyacetone phosphate can be converted into glycerol. This is glycerol. And amino acids are forming the pyruvic acid. Pyruvic acid can be converted into the amino acids. Amino acids the protein. So depend upon the need of the cell. They may work some intermediate products, means here dihydroxyacetone phosphate or the ester coenzyme A or pyruvic acid. Here whatever is shown, they may form by the breakdown of the fats and proteins. At the same time, if the glycerol is required, dihydroxyacetone phosphate directly will go and form that thing in the same way, reverse reaction. Or fatty acids are required, acetyl coenzyme withdrawn from the respiration, it will form. And amino acids are required, pyruvic acid directly will go and form the amino acid by different reactions. That only, it is not only Anabolic, catabolic, it is also anabolic, is there. Amphibolic pathway. I think now you understand properly. If you want to listen once more, please see again the video. Because it is, otherwise if I explain again means it will be lengthy video. So, glycerol will join here, fatty acid will join, how many ATPs are produced also I given, and that complete detail I given. And this is called as a amphibolic pathway. He can create, how he will create? The fatty acids are having 20 carbons, and these are involved by forming the ester that I discussed earlier. Now, 10 glucose molecules are involved in the respiration through the glycolysis and all the things. Out of that one, 5 molecules of the dihydroxyacetone phosphate went to formation of the glycerol, that is the glycerol. And 2 acetyl CoA has gone to the formation of the fatty acids, or 3 or something. And 2 pyruvic acids has gone to the formation of this thing. Finally, how many ATPs will be produced? Very simple. Just you calculate like this. If he gives such type of problem, then you will throw the paper there only. You have to prepare concept I given, develop it in your brain. I told how the questions will come also. He tell glycerol is coming like that, but 10 sucrose molecules, after that one, 4 dihydroxyacetone phosphates are withdrawn for the formation of the glycerol. How many will be produced? Here, from here, how many? 20. 20 into 4 will be less, 80. 10 will produce 380 plus 120. Over it is. Very simple. That is the thing. Now, I think, and one more he has given, very blunder he has given. That is, in the textbook, Krebs cycle he has given acetyl coenzyme A. It used to give out the carbon dioxide, 2 carbon dioxide, because acetyl coenzyme A contains 2 carbon, 2 carbons will go in the form of the carbon dioxide. Now, he has given H2O is coming out from the Krebs cycle. It is wrong. From where it will come? Electron transport system, which is present in the inner membrane. But here he is showing H2O is coming from the Krebs cycle. That is the thing, but in the textbook it is like that. Just I want to show you. Now, respiratory coefficient. What is this? Respiratory coefficient means that ratio which is present between the volume of the carbon dioxide involved and volume of the oxygen consumed. In the aerobic respiration will be done in the presence of the oxygen, all of you know. And the complete oxidation of the carbon dioxide, the glucose or any substrate will produce the carbon dioxide. So how many carbon dioxide are released, and for that one how many oxygen are taken? That is called as respiratory quotient. That is, the RQ is volume of the carbon dioxide evolved during the respiration, and for that purpose how many O2 is consumed, volume of the O2 consumed. This is the thing. In this one, what is the importance? Because based on this one, which food you are digested or not, something, the respiratory coefficient based on that one, what happened, the doctors it will be useful. Now, for glucose, whenever we are seeing here, glucose C6H12O6 plus 6H2O, it gives 6CO2 plus 6H2O plus energy, that all of you know. But the thing is, C6H12O6, 6H2O, here how many are there? Carbon dioxide are coming, 6 carbon dioxides are coming, and 6 carbon dioxide, how many oxygen? Not H2O, it is O2. How many O2 it has taken? 6. So 6 O2 it has taken, and next it is 1. So 6 by 6 is the 1. So when the carbohydrates are involved in the respiration, the RQ will be the 1, because carbohydrates will be finally converted into the glucose only, and it will involve in the respiration in the different stages. Now, this is the thing. Now, one more fatty acid, that is triplamidine. Triplamidine, it is the equation he has given. 2, that is in brackets C55 H98O6. It is a complex complex of fatty acid, because in the living that what is micro biomolecules is read, simple fatty acid contain 19 only, but it is having so many. It is a complex. That is also you will get the doubt why he has given. Okay, it's all right. And 145 OT it has taken. It given 102 carbon dioxide, 98 water, and the energy. So carbon dioxide it given 102, and oxygen it has taken 145. So 0.7 RQ it is having. Now if you see the proteins, it will be the 0.9. He has not given because so many amino acids and all the things. Only the thing is 0.9. What it indicates? Here, proteins and fats, when they are involving in the respiration, RQ will be less than 1. RQ is less than 1. And one more thing, he has not given that, but it is important. The organic acids, when they involve, because organic acids he has given in the starting of the respiration chapter. Rarely organic acids also will work as the food substrate, he has given the sentence. If that organic acid involves, that will be the above 1, the RQ. And next will be the one more he has given, that is in general it will be operated in this way, not in this way, because it is theoretical and assumptional, but it is the actual thing happening in the respiration. So whenever it is operating like this, it is always more than 1 RQ. It is given in the last paragraph. Remember, it is most important for me. Such type of points only you will catch. Generally, what is the RQ of the living organism when it is in the living stage? Generally, it is a little bit 1 to above 1. Why? Because they are breakdown and involving and all the things. So many things are taking. Because of that one, it will be RQ will be in the living organisms generally more than 1. This is most important. Now, in the previous one video I had made, that short video, you see, this is the 6CO2, 6H2O. It is producing. It given in the starting also. Now I am asking, go and see that short video. How many water molecules are producing? This is the 10 plus 2, 12 water molecules are produced in the electron transport system. How it will be 6? That to explain in the whatever the electron transport system. Please see that one. Because here it is 6 only showing, but coming in the electron transport system 12, and glycolysis 2 are coming, and consumption whenever he is giving in the Krebs cycle under that one, 2 water molecules are consumed, then it will become 2, that is 4 minus Krebs cycle 1 means 2 are using 2 pyruvic acid 4, so 12 minus 4, 14 minus 4, that is the 10 molecules are producing according to that one. Why it is, how it is, go and discuss with the teachers. Okay, now or otherwise you see my video. Now we see here, these are the things which are most important for the NEET, and he can give the so many problems also from this one. And if you read, listen all the things properly, you are able to do and answer all the things. Concept orientation I given. Now the thing is, if you like this video, please like it, And next, I am going to start next video with principles of the inheritance, followed by next will be the molecular basis of inheritance, and next will be the biotechnology, which are most important all the things. So second year I will start with principles of the inheritance in the next video within 3 days. So please subscribe the channel if you want to see all the things discussion like this. If you want to see, subscribe the channel. Thank you.